And I've been
Now you've done it.
I know, really. I've been in North Carolina about 30 years, so I say I'm from North Carolina at this point, and I'm a friend of James.
Perfect. Well, welcome all you new timers. For everybody else, thanks for coming back. Oh, one more. Oh, wow.
I'm a diver. All my friends and I are all on the Huntsville dive team, so local boss as well. It's fun. So wanted to join and find out more.
Very cool. Well, welcome everyone. All right. So yes, we're getting a very diverse section of people here. So I guys want to welcome you again, and I'm going to introduce now our speaker for today, and it is Dr. Jennifer Peteya. Did I do it right, Jenny? Okay. She's going to talk to us about filling in the dinosaur coloring book. This should be a great talk on the new field of color in fossils. So I'm going to turn it over to you, Jenny. And
Great, thank you, Linda, and thanks everybody for having me and for coming. I'm in Pennsylvania, so it is really cold here, and it snowed really bad. So, like, my apartment complex is covered in snow. Hopefully, you guys don't have snow. You're south; it should be fine. So, little bit of a switch up. So instead of talking exclusively about dinosaurs, I wanted to make my talk a little bit more relevant to you guys. So most of it is going to be about filling in the dinosaur coloring book, but I've added some North Carolina fossil stuff to it as well. Just real quick, a little bit about myself. I am currently an assistant professor in the biology department at Bloomsburg University, which is now called Commonwealth University of Bloomsburg. So I'm in Pennsylvania. I'm from Ohio. I did my undergraduate degree in geology at Mount Union College in Alliance, Ohio. I did my master's in earth sciences, looking at trilobite guts at The Ohio State University. And I did my PhD on what I'm going to talk to you about today at the University of Akron. So I'm excited. I'm happy to be here, and thanks again for having me. So this is filling in the fossil coloring book, not just dinosaurs. If I can get this to work. There it goes. So the world today is very, very colorful, and animals are a huge contributing factor to all of those bright and beautiful colors that we can see in the natural world. You have some duller colors in certain animals, like mammals, for example. You never see a blue cat unless it's been dyed by some horrible person. You always see like black cats, brown cats, orange, red cats, etc. But a lot of other animals are capable of producing some amazing colors. For example, we have mallard ducks here, this beautiful spot on the female and down here on the male. Those are iridescent colors. So depending on how you look at them, they might appear blue, they might appear black, they might appear violet. Same with the male's head here, this beautiful green color that you just don't see in mammals. The flamboyant cuttlefish has an amazing array of colors that they use to surprise their prey and for attracting mates. Geckos, amphibians like the poison dart frogs, and we see them in sea urchins as well, these bright, beautiful purples and other colors. So the world is a very colorful place. And we therefore, since colors exist today and lots of animals have lots of colors today, we can assume that the ancient world, the prehistoric world, extinct animals had similar colorations and color patterns as well. But we never thought that we'd be able to determine that. So starting off, I just want to start with how animals make color, using birds specifically as an example, because that's what I worked the most on. So colors in the modern world are produced by two main mechanisms. The first one is by pigments. This is how mammals, for example, make color. This is how lots of birds and other animals as well make color. So pigments exist to absorb certain wavelengths of light and reflect others. So for example, the sun. The sun creates all of the different colors in the visible wavelength that we can see. So that's bright white light. That light, let's say we have a cardinal, a northern cardinal like this little male here. he has a pigment in his feathers. Specifically, it's a carotenoid called astaxanthin that produces this red color. So under the keratin of his feathers, he has pigment granules made up of that carotenoid. When sunlight hits his feathers, certain wavelengths of colors are absorbed and red bounces off of it. It's reflected. So any violets, blues, yellows, oranges, etc., get absorbed by that carotenoid pigment, and red is not absorbed. So it bounces off, it reflects off, and we see this beautiful bird as red in color. That's just one example. Melanin is the main pigment that I'm going to talk about today. Melanin also works in a similar way by absorbing, depending on the type of melanin, absorbing most of these colors that the sun produces so that we see black or so that we see red-brown. So it works in a similar way. We also have animals that produce what we call structural colors. When it comes to structural colors, these aren't just pigments. They might involve pigments, but it's not just pigments interacting with sunlight. You have interactions between different materials, including pigments, inside of the animal's integument, whether it's the skin of a lizard or the feathers, in this case, of a bird. So there's two different types of structural colors in birds. We have non-iridescent structural colors and we have iridescent structural colors. Non-iridescent structural colors, you have a pigment base, usually that is a black melanin, like eumelanin, which we'll talk about, under the feather structure. Feathers are mostly made up of keratin, the same or very similar protein to what's in your hair and your fingernails, just a little bit different in feathers. So you have the keratin of the feather followed by a layer of black melanin. So when the sunlight goes through that keratin, it hits this weird spongy type layer of keratin and also the black melanin underneath that. So the melanin absorbs most of the sunlight that comes through. In the case of the indigo bunting here, it's absorbing red, yellow, green, etc. But in the case of our indigo bunting, the spongy layer of keratin is arranged in such a way that that sunlight hits air spaces between the keratin and bounces back blue color. So that's why we see the indigo bunting as blue. It doesn't have any blue pigments in it. It's just this weird interaction between light reflecting off of keratin and melanin. So that's non-iridescent structural colors. Iridescence means that if you change your viewing angle compared to the feather is going to change color. So for example, the mallard that I showed earlier, his green head, you might see as very bright green looking at it straight on, but if you look at it from an angle, it might look black or it might look brown. So the color changes depending on your angle to the feathers. So it works in kind of a similar way to non-iridescent structural colors. You still have light bouncing off of pigment and light bouncing off of keratin. So in our hummingbird here, hummingbirds are one of the most excellent examples, and we'll see another example here in a moment. But depending on your viewing angle, the bouncing of the light off of the keratin layer or the absorption of the melanin layer is different. So it's reflecting violet at certain angles, it's reflecting blue at certain angles, and green at other angles. So it's layers of aligned melanin granules that we call melanosomes and layers of keratin that interact in such a way to change the color of the light depending on how you're viewing it. So those are structural colors. Pigments just absorb light. Structural colors, you have a lot more complex interactions between the keratin of the feather and the pigments that are associated with the feather, usually melanin. So melanin was my focus for my PhD. It is the most universal pigment. Currently, as far as I know, it occurs in every major animal taxon out there. When I was a PhD student, for like half the time I was a PhD student, no one had found it in spiders. We took a spider, stuck it under a Raman microscope, and we actually found melanin in spiders. So every major animal taxon now has melanin that we know of. So melanins are the most ubiquitous pigments. They occur naturally in lots and lots of different types of animals. The main melanins that are associated with color are called eumelanin and pheomelanin. Eumelanin is the most common. I have my pretty calico here as an example. Her name's Adria. She's 13 now. So Adria is a beautiful canvas of color. She has black around her nose here and around her face. That black is produced by eumelanin. Eumelanin can also produce dark brown colors. So black and dark brown is eumelanin. As I said before, eumelanin also heavily contributes to structural colors in other animals, not mammals. Pheomelanin, so the orange colored spot around her eye here, kind of this rusty red orange color, that is produced by pheomelanin. So pheomelanin produces rusty red colors as well as some buff yellow or dull yellow type colors as well. Then, of course, she has white on her face. She's got white on her cute little paws. White, you don't have any melanin. You don't have any other types of pigments. It's just white. So if the sun's reflecting or if the sun hits her paw, none of that light is being absorbed. It all bounces off. You see it as white. So no pigment there. What's interesting is that in most vertebrates, eumelanin and pheomelanin are both housed in organelles that we call melanosomes. If you're not familiar with the term organelles, they're just little tiny subsets of little tiny compartments inside of cells that do things. So the mitochondria, for example, is the classic example. It's the powerhouse of the cell. So in this case, our organelle is melanosomes. Melanosomes house melanin. They house some percentage of eumelanin and some percentage of pheomelanin. This is what they look like. So I took, not this one, this one's from the internet. I took a mud puppy carcass and sectioned some of the skin and found these little tiny microbodies. The scale bar here is 1 micron, so they're very, very tiny. These are the melanosomes. So there's melanin inside of there that is contributing to the dark green color spots on the mud puppy. So that's essentially what they look like. What is really cool is that in mammals, to a lesser degree, and in birds, the melanosome shape actually correlates to color in the animal. So this is an example that I modified from a paper called Lee et al. 2012, where they looked at different types of bird melanosomes under a scanning electron microscope, so a very high-powered microscope. And they saw these tiny meatball-shaped melanosomes in rusty red colors. So we would expect those to have a very high amount of pheomelanin. They saw these pill-shaped melanosomes, so thinner and longer than what we saw in these, in black cormorant feathers. They saw still pill-shaped melanosomes, but kind of bigger and fatter in gray cockatoo feathers. And in iridescent feathers, they found very highly elongate pill-shaped melanosomes that were aligned end-to-end in sheets. So that's why I have kind of this random orientation in these guys, which you can also see in the SEM images, and you can see the end-to-end orientation in the iridescent feathers from this duck. So small meatball-shaped melanosomes, these kind of rounded melanosomes, are associated with red-brown colors, pill-shaped melanosomes in black, weirder pill-shaped melanosomes, fatter pill-shaped melanosomes in gray, and then very long end-to-end oriented in iridescent feathers. These aren't the only shapes that exist. So we have color correlation here. If you look at hummingbird feathers, hummingbirds are really strange. So they have iridescent feathers. This is Anna's hummingbird, a male. And you can see kind of as the GIF goes, if he looks directly, almost directly at you, his feathers are bright pink. And if he looks to the side, they're kind of a duller green-brown color. So very highly iridescent. The reason for that, oh, my computer's not plugged in. The reason for that is because they have these weird kind of platelet, almost pancake-shaped melanosomes. So that weird melanosome shape contributes to this bright color, this highly saturated iridescent color that you can see in these animals. Give me 1 second, I'm just gonna switch my computer cord here. Before it dies, sorry about that. So these weird pancake melanosomes we find in hummingbirds and hummingbirds only so far in the modern world. We'll come back to these guys. So what about dinosaurs? Dinosaur studies really got started in the early 1800s, even though dinosaurs had been found for hundreds of years beforehand. But scientists never thought that we would be able to determine the colors of these animals. So we have the classic examples from Charles Knight down here, where the dinosaurs are very, very dully colored. We have the example from Yale Peabody Museum, where the dinosaurs are very dully colored. They're all drab greens, grays, browns. Nobody likes a non-colorful dinosaur. That kind of went up into the 90s. So this is an example from Lost World Jurassic Park specifically, but you look at Jurassic Park movies and you don't really get much color in the dinosaurs until Jurassic Park 3, and even then you barely get any color. So we don't really have dinosaurs represented in popular media with vibrant, beautiful colors until the 2000s. So can we actually color dinosaurs? The cool thing about science is that it's ever changing. And as technological advances happen, and we get cooler and cooler toys to play with, the science tends to get cooler and cooler, I think anyway. So the scanning electron microscope, very powerful microscope, can look at very tiny things using electrons. And it was invented in the mid-1900s. And pretty much from the get-go, paleontologists were like, hey, let's scan things with this and see what we can find. So this is a paper from 1995 where Davis and Derek Briggs, Paul Davis and Derek Briggs went in and took a look at some fossilized feathers. And what they found under the scanning electron microscope was these weird little bodies, kind of like this. And they thought, okay, these have to be bacteria. They're probably fossilized bacteria associated with the decaying carcass. And that was that for a long time. Derek Briggs moved to Yale. He's now at Yale, and he ended up with a student, Jakob Vinther, who's now at the University of Bristol. In 2008, Jakob Vinther took a much closer look at fossilized feathers using a scanning electron microscope and found the same microbodies as Derek Briggs had found several years earlier. So this is actually the feather that he looked at. It's from the Messel pit in Germany. And he found microbodies just like those in these dark stripes on this feather. This one, where you don't see any microbodies, is from these lighter stripes. So kind of makes you wonder, like, if it's decay bacteria, why don't you have bacteria covering this entire feather? And that was one of the questions he wanted to answer. So he actually figured out that these are probably melanosomes original to that feather and not just bacteria. And this spawned a whole revolution in the study of dinosaurs. The new field of paleocolor, which I have been very fortunate to be a part of. So this was kind of the paper that led to all of that. So I have to mention this. This is what Jakob Vinther kind of looked at in this first initial paper. Why are they not bacteria? Why are they melanosomes? And like I said, those microbodies, these little tiny structures, only exist in the dark parts of the feather where you would expect to find melanin. They are similar, most similar to melanosomes in size and shape. Bacteria tend to be quite a bit bigger than this. And then eumelanin is very resistant to degradation. We know that now from lots of different experiments. So chances are these are melanosomes. We now have a lot more evidence to back up the idea that these are melanosomes than Vinther had in 2008. If that's something you want to ask about, feel free to go for it. I'm going to show you some chemical evidence here in a moment. So the next study. Like I said, 2008, Jakob Vinther's paper kind of exploded this cool idea that we could color dinosaurs. And the first study to really do that was Lee et al. 2010, which my PhD advisor, Matthew Shockey, is on. So what they did, they looked at Vinther's paper, they were like, okay, melanosomes are preserved. Do dinosaur feather melanosomes have the same color to shape correlation that bird feather melanosomes have? And so Anchiornis was the first dinosaur to be colored based off of this idea. So this is Anchiornis huxleyi. It's a dinosaur from China. And this is the head. You can see this black stuff around the head. That's feather preservation. You can see it a lot better on the wings here. So these are the arm bones. You've got wings coming off of there. Chest feathers, and we have some cute little leg feathers here as well. So they took several samples from this animal and ran them under a scanning electron microscope. And they found in most cases that these samples had these little microbodies in them. Some of them are three-dimensionally preserved. Some of them are just impressions of the microbodies that used to be there. They took these, did some statistical analyses on them, and found that indeed these melanosomes are similar in size and shape to the color correlated melanosomes that we see in modern day birds. And so they were able to color this animal kind of like this. You've got these beautiful crown feathers that had those tiny meatball shaped melanosomes in them that they think were red-brown in color. On the wings and the leg feathers, you have kind of these darker spots on the feathers that were probably black. So they would have had the elongate pill-shaped melanosomes, and then kind of the fatter pill-shaped melanosomes for the rest of the body, making most of the animal gray. So this was the first dinosaur to be colored. The white, or one of the first dinosaurs to be colored. We'll get back to Sinosauropteryx. The white here, the fossil doesn't really have feather preservation in those areas, so they assume that it was white because they didn't find any melanosomes there. And there's no dark coloration where the white spots are there. So this animal probably looks something like this. Then in 2012, they found the first evidence of iridescence in a fossil. So this is Microraptor, also a dinosaur from China. And you can see the skull's over here. The tail is over here. It has some beautiful feathers that hadn't been described yet. The wing feathers here, you can see leg feathers here. So beautifully preserved animal. Again, took several samples, ran them under a scanning electron microscope, found extremely long melanosomes, long and thin melanosomes that were aligned end to end, just like we saw in an iridescent duck feather. So they think that this animal looked something like this. It probably had iridescence like a grackle or something like that. The unfortunate thing is with melanosomes, we can only tell that this animal was iridescent. We don't have the keratin there to determine what hue it was. So we don't know if it was blue iridescent, purple iridescent, violet iridescent, or green iridescent. And we may never know. We'll see. So that's the first evidence of iridescence in the fossil record. But wait, there's more. It gets even more fascinating from here. This is Kai Hong. So Kai Hong Jujai was described by Hu et al. In 2018, right as I was finishing my PhD. And it's a seemingly normal Chinese dinosaur. You got the beautiful feather preservation. The skull is here. There's some fan-like tail right here with beautiful feathers all around the body, etc. And so, of course, they took samples of it. Let's find out what color this animal is. And boom, something fascinating happens. You have the fossil samples up here compared to hummingbird melanosomes down here. These fossil melanosomes are that weird pancake shape that we see in modern day hummingbirds and only in modern day hummingbirds, which is so weird. You wouldn't expect to find these in the fossil record unless it was a hummingbird. So what they think is going on here is that Kai Hong had these hummingbird-shaped melanosomes and that they were highly iridescent, just like we see in different types of hummingbirds, and like we saw in the GIF of the Anna's hummingbird. So this is the reconstruction of Kai Hong Jujai. It probably wasn't rainbow. Again, we don't know what the hue is, but it would have had that highly, beautifully saturated iridescence that modern day hummingbirds had in its head and neck feathers, at least. So pretty amazing discovery. So those are only a few examples. There's plenty more, and I'll get to one more here in a moment. We know that melanosomes are preserved in fossil feathers. We've seen it several times now. What about the actual melanin itself? Eumelanin and maybe pheomelanin as well. And the answer is yes, we have found eumelanin at least. So there have been a number of studies now that have found eumelanin. It's not all just based on these cute little SEM images of melanosomes. We have melanin now. The most common technique that is used to find melanin in fossils is called TOF-SIMS, or time of flight secondary ion mass spectrometry. I've done it. It's difficult. It's very expensive and it's hard to interpret. But it does tell us with high certainty that we actually do have melanin, specifically eumelanin, preserved in these fossils. So this is an example from Lindgren et al. 2015, where we have the eye of this fossil bird. You can see the melanosomes from the eye, and then this is the TOF-SIMS readout. Again, kind of hard to interpret, also very expensive. So part of my PhD was to look at a less expensive method to look for melanin in the fossil record. So this is the one of the fossils I got to work on. This is my favorite fossil that I've ever worked on. Unfortunately, he doesn't have a name. His name is currently CUGB P1202. There's several reasons for that. So he is from a group of birds called the Ananiornithines that went extinct 66 million years ago, along with most of the rest of the dinosaurs. They're related to modern day birds, but they have some traits that are very, very different from modern day birds. For example, claws, teeth, some weird things going on with their bones that modern day birds don't have. So I looked at this guy and he's got some cool things going on. So you can see the black skeleton here. He's from China. He's about 123 million years old. Wing feathers. He's got some tail feathers here, including these two elongate tail feathers that are common in Confucius ornis, for example. And he's got some pretty crown feathers here as well. So beautiful feather preservation. He also has some cool things going on with his bones. So this is not an adult animal. It's also not a baby. He had some bones that weren't fused like they are in adult animals, and he has some bones that still showed pitting like they were still growing. So we think that he was a subadult animal, not a baby, but not an adult. The feathers also suggest that he was male. I'll get more into that here in a moment. So I took some samples from his feathers, and again, like the others, ran them under a scanning electron microscope, and we found these highly elongate melanosomes arranged end to end, just like we saw in Microraptor and the iridescent duck feathers. So this is my drawing, my interpretation of what he looked like. He probably had iridescent feathers, just like a modern day grackle or a duck. So kind of a cool animal. We also took these samples and ran them using Raman spectroscopy to try to figure out if melanin was preserved. The nice thing about Raman over TOF Sims is that it's less expensive, it's less damaging to the specimens, and it's pretty easy to use and pretty easy to interpret for the most part. So we were hoping that this would be a good alternative to TOF Sims. What Raman does is you take your sample, you put it on a slide here, and it shoots a laser down at your sample, excites the molecules on the surface of the sample, and then reads what's going on with those molecules. And then based off of the readout, you can interpret what chemicals or what molecules are present in your sample. So what this is the readout from CUGB P1202. These are the fossil samples down here. These are modern samples that I compared that to. So we've got chicken, red-winged blackbird, house wren, Cooper's hawk, mallard, and turkey, wild turkey. These are a classic eumelanin signature. So that dark black melanin. We have a smaller peak here, and we have a larger peak here. Very common or very signature for eumelanin. We also see that in our fossil samples. So all of the fossil samples that I took had those two characteristic peaks. I will say that there is a caveat to this method for melanin. These two peaks are very annoyingly similar to carbon peaks. So Raman spectroscopy isn't the greatest technique we found for looking at eumelanin specifically, but it's good for other things. For example, the fossils that Linda and I are going to be looking at, I hope. We will see. But we have that nice correlation that tells us that melanin might be present in these samples. So we know that eumelanin is preserved in the fossil record. There are several studies that say that using different techniques. It makes sense because eumelanin is very difficult to break down. And it's an extremely complex molecule. But what about pheomelanin? Pheomelanin is a little bit more contentious in the fossil record. It's tricky because some people think that it breaks down faster. Some people think that it doesn't. And experiments that have been done on modern day melanin do suggest that pheomelanin might not preserve as well. There are two studies that I know of that have really kind of gone into this pheomelanin question in fossils. One is Brown et al. 2017. They looked at this gorgeous fossil. This is Borealopelta, which is a nodosaur, so a plant-eating dinosaur related to Ankylosaurus, those big club-tailed dinosaurs. Only nodosaurs don't have the club tails. And it was found in a coal mine in Canada, and it's currently housed at the Royal Tyrrell Museum in Alberta. They took samples from the skin of this animal, and they think that they found markers, biomarkers for pheomelanin. Maybe they did. The problem is that the markers that they used can also indicate other things are present. So for example, I can't remember if they actually did this in this paper, but for example, sulfur. Sulfur is found in a lot of things. It's also a marker for pheomelanin. So it's a little bit questionable. This study, on the other hand, just came out. This is by Tiffany Slater et al. And she found biomarkers, again, for pheomelanin in several different fossils. So what this is showing markers for eumelanin in these fossils and markers for pheomelanin in these fossils. And she used HPLC, which is very commonly used to extract melanin in modern feathers. And hopefully she did find pheomelanin. It's a very interesting study. She thinks that she has pheomelanin in a bird called Confucius ornis from China, a dinosaur called Sinornithosaurus, and some frog specimens as well. So it's possible that pheomelanin is preserved in the fossil record. We just need more evidence. Eumelanin at least. So the next question. We know these animals had color. We know some of them were very brightly colored. Some of them were extravagant. What did they use them for? Why were they colored in the first place? For that, we have to compare them to modern day animals. For the dinosaurs, at least, we compare them to birds. So we have no reason to assume that they didn't use their coloration, the colors of their feathers, for the same, no reason to not assume that they didn't use their coloration for the same reasons as modern day birds. So for example, with Anchiornis, you have these beautiful, what they call spangles in the paper, these kind of spots on the feathers. Modern day birds like nightjars, for example, use those spots to camouflage themselves. Nightjars are night hunters. They camouflage themselves against trees during the day so that they could sleep, and also to help ambush their prey. Anchiornis may have done the same kind of thing with its feathers. This is a carnivorous dinosaur. Maybe it did the same thing. However, you can't rule out mate attraction. So they might have used this to woo the ladies. You've got this pretty red crest right here and these pretty little cheek feathers. Maybe the females liked them. Who knows? So they might have been used for camouflage. They might have been used for mate attraction. Iridescence. So we saw that in CUGB P1202, Kai Hong, extremely iridescent, whoops, and Microraptor, and a couple of others now. That is almost exclusively used, as far as we know, in modern day birds to attract mates. Iridescence's uses in other aspects of their life haven't been studied very well, mostly just mate attraction. So for example, we looked at Anna's hummingbird. Anna's hummingbird, only the males have iridescence, the females do not. That is the case for most birds, most modern day bird species. The male has the iridescence, the female does not. So it's possible that it was the same for Microraptor and Kai Hong and CUGB P1202, that they were using that iridescent color to attract mates. The other cool thing, Kai Hong is also the oldest example of iridescence that is known in the fossil record. And it had, again, the weird plate-shaped, pancake-shaped melanosomes that you see in hummingbirds. So those traits are at least 161 million years, way older than we would have expected, which is really, really cool. Then we have my baby, CUGB P1202. His iridescence is pretty significant for the type of bird that he is. So again, he's an enaniornithine bird. They went extinct 66 million years ago. They're related to modern birds, but not too closely. So we know based off of his melanosomes that he had iridescent feathers. He also had these beautiful elongate tail feathers, which most modern day birds, if you have elongate tail feathers like a peacock or like a whydah or something like that, they're limited to the males. So this may be a male trait. And again, iridescent is usually only limited to males in modern birds as well. So he was probably male. He was also a subadult, so he wasn't fully grown yet. So he has these sexually selected male traits, iridescent and ornate tail feathers, but he's not yet an adult. In modern day birds, most birds don't get their ornate feathers. They stay dull in coloration. They don't get the long tail feathers until they have reached full sexual maturity. This guy was not fully mature yet, unlike modern day birds. So this adds to several other lines of evidence that enantiornithine birds developed sexually before they became adults. So they were reproducing before they were fully grown, unlike modern birds, which blows my mind. It's so cool. So anyway, he's my favorite. The next question, can we color all dinosaurs? Unfortunately, the answer is no. You may have noticed that all of the dinosaurs that I have talked about are either birds or very closely related to birds. We have looked at skin and feathers and proto-feathers from other dinosaurs that aren't very closely related to birds. Anchiornis was one of the first dinosaurs that was described with color. So was Sinosauropteryx. These two papers came out within weeks of each other from different authors. Sinosauropteryx is not closely related to birds. It had kind of these little primitive feather-like structures on it. This is the tail of Sinosauropteryx here, and you can see kind of these dark feather-like structures. When they looked at this animal, they found kind of these meatball-shaped melanosomes, and they assumed, based off of modern day birds, that these meatball-shaped melanosomes correlated with red-brown color. Okay, that's fine. It's 2010. We don't know a lot yet. Then we started looking at other dinosaurs. This is Bipausaurus, which is a Chinese dinosaur related to Therizinosaurus, so the big long clawed dinosaurs. Again, we have feather-like preservation here, meatball-shaped melanosomes. And we see that in other dinosaurs too. Psittacosaurus, for example, this is Sinosauropteryx. So most of, or all of the dinosaurs so far that we are finding that have longer melanosomes and more variation in melanosome shape and size are what we call the Pentaraptora. So they're very closely related to birds and include birds. What this is an image that I made, a cladogram that I made, showing melanosome size and shape variation between different types of animals. So we have our, from here down, these are Pentaraptora, so the types of dinosaurs that include birds and are related to birds. Then up here we have two different types of dinosaurs, Psittacosaurus, which was a plant eater, and Sinosauropteryx. This is a pterosaur, like you probably know Pterodactylus or Pteranodon. Crocodiles, ichthyosaurs, so extinct group of marine reptiles, different types of reptiles. Here's a mammal, and then amphibians, fish, and lampreys. This shows you the type of integument associated with it. So these weird things are skin, hair for the mammal, and then kind of these pycnofibers or protofeathers for pterosaurs and some of the more primitive dinosaurs, and then real feathers for the Pentaraptorans. Then this, on this side, shows you all the variations in melanosome shape that we have found. So if you look at modern day birds, for example, down here, these are modern day bird melanosomes and the colors they are associated with. So small meatball shaped melanosomes for red-brown, pill shaped for black, wider pill shaped for gray. This is penguin type. Penguins have weird melanosomes. And then these purple ones are iridescent colors. So iridescence comes in some high variation. These ones that aren't colored in are fossils that are associated with iridescence so far. Or sorry, that are associated with bird fossils so far. So fossil melanosomes here. This is anaornithines like my birds, UGBP1202. The variation that they have is pretty high in size and shape. All of this is scaled to real melanosomes. Microraptor and related dinosaurs here. So still size and shape variation. If we go up to mammals, we can see some variation, obviously not as much, but you've still got the long black melanosomes, the shorter red-brown melanosomes, etc. You don't see a lot of variation in most of these other groups. So Psittacosaurus had small meatball-shaped melanosomes. Sinosauropteryx and several of the other dinosaurs that have been studied have short meatball-shaped melanosomes. Same with lampreys, fish, amphibians, other types of reptiles. You see no real variation between melanosome size and shape, and none of it is correlated with color. The weird one here is pterosaurs. There was a paper that came out 2 years ago now, I think, on a pterosaur called Tupindactylus, showing that it did have long melanosomes just like we see in birds. So pterosaurs are a weird one that need more study. So basically what I'm trying to say here is we can't color every dinosaur because they don't all have the variation that we see in modern day birds. Hopefully that made sense. So that's it for dinosaurs. I want to switch gears here and briefly discuss North Carolina fossils, because that's what I'm starting to work on now, and what I'm involved with Linda with here. And it's relevant to you guys because you're in North Carolina. So dinosaurs aren't the only things that are being colored in the fossil record. We do have evidence of color in other animals, specifically invertebrates. So we have colors in trilobites, those cute little marine arthropods. They haven't been heavily studied yet. That's something else I'm hoping to look at. But I want to show you echinoderms. So echinoderms include starfish, baskets or brittle stars, sea cucumbers, and these cool animals called crinoids. So crinoids, you can kind of think of them as like starfish with stalks. So they have, most of them have this kind of cool stalk attached to them. So these are animals. They're also called sea lilies, but they are not in any way related to plants. What you're seeing here, this is an example from Ohio State, right before I started my master's, where Chrissy O'Malley and Dr. Osich looked at, saw these cool crinoids from Indiana. These are Mississippian in age. And they noticed, hey, these crinoids are on the same slab, but this one's kind of white, this one's kind of black, and there's some gray purple ones in here too. Maybe something weird with the color is going on here. And they found biomarkers for quinones. Quinones are another type of pigment that you don't really find in like mammals or in birds, but you do find in a lot of invertebrates. So most modern day echinoderms have quinones in them that are coloring them. So that's probably what's contributing to these color differences here. This is another paper from Wolkenstein, also looking at color in crinoids. And you can see kind of there's pink color going on in this crinoid here. You can see a little bit better in the cross section here. They also, using a different technique, found biomarkers for quinones. So we have evidence of pigments and color in other animals as well, not just dinosaurs. Which brings me to North Carolina. North Carolina has colored fossils, which I'm sure Linda has talked to you about already. You guys have an amazing array of invertebrate fossils that you can just go to the beach and find, which is really, really cool. And some of these might actually contain remnants of their original color. So how this worked was I went to, with my friends, to Topsail Beach, North Topsail Beach, in 2018, and had no idea that they had dredged, had no idea that there were fossils on the beach, and ended up finding a bunch of these cool sea biscuits, which are, of course, also echinoderms related to these crinoids. So this is Hemiptegus, one of the ones that I collected in 2018, and it's got some weird color thing going on here in the ambulacra. So some of them have kind of this purple violet color, and I was scratching my head. I found more in 2019 and was really wondering, hey, I wonder if anyone's done anything on this. So how this happened was you had an offshore dredging project to revamp the beach in 2015, and it stirred up thousands and thousands of fossils from the riverbed formation offshore. So the riverbed formation is a Oligocene. These fossils are about 30 million years old. Hundreds of these fossils appear to have some sort of color patterning, including our Hemiptegus here. So I was curious if anyone had done anything about this. I looked it up on the internet and I found Linda. So Linda has reported on these fossils at a few GSA conferences. And I was like, has anyone done chemistry on this? So on a whim, I emailed her. No, nobody's done chemistry on this yet. So Linda and I are now friends. We are actually going to be working on these starting this semester. So I'm going to get time on Lock Haven's campus to actually take a look at these chemically, and we'll hopefully be able to write these up, which will be really cool. So if you've collected these fossils or if you've listened to Linda's talks, you've known the Hemiptegus have color patterning. You have color patterning in bivalves. So these kind of cool orange looking stripes here have not been described. You've got kind of this red violet color patterning in barnacles. There's some cool pink things going on with the brittle stars. Lots of amazing examples of color in these animals that we're hoping to figure out if it's original color or if it's diagenetic. So basically mineral precipitates. I don't think it is. So do they contain original pigments and how are they preserved? Stay tuned, we're gonna find out. I visited Linda and stole some of her fossils from her really cool collection in November, and we're gonna get started on them soon. So what I'm going to do with these is I'm going to take samples of some of her fossils and some of my fossils and look at them under Raman spectroscopy. So Raman, I said, doesn't work terribly well for melanin, but it does work pretty well for other pigments, including quinones. So we have evidence of quinone pigments in other echinoderms. Maybe we have quinones in these guys as well. Modern day sea urchins, we do know have quinones, specifically quinones called echinochrome and spinochrome. So we're gonna see if we can find those by looking at the fossils under Raman and comparing them to modern day relatives, specifically Mauritia from the Philippines. I'm also hoping that I can get some quinone standards to compare them to as well. We'll see. So we think that these colors are original to the organisms. We're hoping that we will find quinones. We'll see. We're also going to do some other experiments with these as well. As you notice, so this is modern day Mauritia. This Mauritia has color, these don't. The difference here is the skin is preserved on this animal, whereas this is just the test underneath. So this is the calcium carbonate underneath that skin that makes them nice and hard. Obviously, we don't have skin here. So how is color preserved on the test here when it's not preserved on the test and its close relatives? The test isn't producing that color. So how is it preserved? So we'll see if we can do some experiments here trying to figure out how this is preserved here as well. And I know Linda has some hypotheses on this if you want to talk to her about it. So that's the future. Hopefully this will go well and we will get some cool things out of this. I really hope. So last thing I just want to mention, again, thank you for having me. Fossil color studies are extraordinarily important. It might not seem important to discover what the color of a dinosaur was or to figure out if pigments are preserved, but it actually is. So what it tells us is something about the lives of these animals that went extinct millions upon millions of years ago. And it tells us about their anatomy, their physiology, how they might have interacted with other members of their species, other members of other species, how they might have interacted with their environments, and how similar they were to their modern day relatives. There's a lot of really cool things that are going on, and hopefully we can continue to figure these things out. And with that, thank you. This is a cool photo from November in Holden Beach. I cheated, but there could be color in the fossils that are currently washing up at Holden Beach too. We will see. Anyway, thank you. Does anyone have any questions?
If anybody has any questions, if you come stand up here, you'll be able to ask her. So come on up here and so she can hear you.
Hopefully I didn't go too long.
Hi there, thank you for that wonderful presentation.
Hi.
Much enjoyed it. Quick question then, has there been any studies done on other types of marine life, in particular, say, fish and cephalopods, including ammonites?
Yes. There's this really cool animal. I don't know if you've heard of it, called the Tully monstrum.
Oh yeah, that's the state fossil of my hometown in Illinois, yeah.
Oh, there you go. So you know the Tully monster. There has been, Tully monster is one of the oldest fossils that has been studied for color. They found melanosomes, it's Clements et al., Thomas Clements wrote the paper. They found melanosomes in its eyes and melanin, which is really, really cool. So you have this Carboniferous age fossil with melanin and melanosomes. There's also been some work done on teleost fish that are about the same age, where they found melanosomes as well. As for other colors, I mentioned the crinoids, trilobites. So those cute little marine arthropods have color patterning. They haven't been heavily studied yet. So we'll see what's going on there as well.
Cool, thank you very much, I appreciate it.
Yeah, thank you.
Anybody else? You got her live now. Come and ask if you got any questions. No. Okay, Jenny, looks like that was it.
Cool.
Thank you very much. We're gonna give a round of applause.
Thank you.
And we're gonna end up having a break and then a meeting.